Okay, so we're going to be continuing on with our genetic notes. So now today we actually going to learn about um, Punnett, the other Punnett squares with incomplete dominance, codominance, and as well as getting ready to introduce you to pedigrees. So today we're going to understand incomplete dominance, codominance, multiple alleles, as well as polygenetic traits. So complex of patterns of inheritance. So again, you all did pretty well with the Punnett squares. But many inheritance patterns are more complex than what we did with the Punnett square, though, just with the regular genes and the dominant and recessives. Not all traits are actually just dominant and recessive. And this little chart right here is a pedigree. We will actually learn this in our next set of notes. So incomplete dominance. So this is when one allele is not completely dominant over another. So basically, I call this the mixed kid. Okay? So let's look here at this picture. We have a fork and we have a spoon, and they had a baby, and that's called a who? A spork. Yes. All right, we have a red flower, we have a white flower. They had babies, the babies came out pink. So do I see the red? No. Do I see the white? No, I see a mixture. So incomplete is basically like mix. So the heterozygous phenotype is between the homozygous phenotypes. So it's like right there in the middle, like we don't know what it is. So let's just say this is red and this is white. So the kids, yes, they're going to come like this, and it's just kind of showing in between. I know the letters are a little bit different, but this is incomplete. So incomplete, I just think of mixing, okay? So here is an incomplete example, a dominant example of a snapdragon. So a red and white flower makes a pink flower. But when they had the babies, they had one red, they had two pink, and one white. Okay. Now, this can happen in adults and happens in humans, and not just adults, kids too. Just basically humans. It's sickle cell anemia. This is a disease that is prominent in Black Americans whose families originated in Africa, and white Americans whose families originated inside the Mediterranean seas. So, sickle cell is an issue on the genotype. So, if a sickle cell gene is inherited from one parent, you carry the trait. So, you see how it's only on one letter. There's a little C on that letter. That means you carry the trait. You do not have sickle cell. If you had sickle cell, there will be another little C like this there. You are actually resistant to malaria because malaria cannot get on that. So that's the only good thing about sickle cell. But honey, sickle cell is not really that good. If you have sickle cell, again, you had to get it from both parents. That's the only way you can get it right there. You would have two carriers on it. So now what causes sickle cell? So an oxygen carrying protein called hemoglobin differs from the normal hemoglobin. So look at the shape. This is ours. I told you ours is our regular normal blood cells is like bumper cars. So we go through the body, bump, 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 get from the head to the toes. But once you like this, it can cause it to clot, right? It's like hooks. So they can get stuck on each other, which just causes body pains. And because of this, they're shaped like half moons, but it can slow the blood flow and it can back up, which causes pain, right? Because it's just a blockage. It's a hook. You ever seen the little monkeys that was in the jar? Maybe I'm just a little getting too old. It was a little red thing and the monkeys are in there and you shake it up and you pour it out and see how many monkeys are connected at the tail. That's basically what happens here. It cannot flow. Sickle cells have shorter lifespan than normal red blood cells, which causes them being anemic and having anemia, which can be very deadly because if they get cut or something like that, they can get a blood clot or they can end up the blood doesn't last as long. But now let's talk about codominance. So incomplete dominance, you see, remember, that is just going to be mixed. Codominance, both of them are wanting to be dominant. Like, no, you ain't, no. If I said it, we both say it, right? So if you look here, both alleles are expressed. So if this flower is supposed to be pink, well, we saw pink and white. We see a cow, and he brown and white. He didn't know if he wanted to be white or he didn't know he wanted to be brown. But hey, he's both of them. That is codominance. So let me show you an example of codominance, okay? So we have here, the R is for red flowers, okay? And then we have a W for white flowers. Now, of course, we was always pretty, and I showed you in the regular Punnett squares that this right here could just be a dominant and a recessive, but both of these are dominant alleles. So red is dominant and then white is dominant. So let's go ahead and do the box. Here's our red, here's our W. So this is going to be RW. I'm not sure if I have another W, but if it pops up. And then RW, RW. So all of these come out polka dot. This is what I call polka dot, okay, because it shows both of the colors. Now, codominance does happen in humans, though. So we also have incomplete, which was obviously the sickle cell, and now we have codominance. And this is when it comes with the phenotypes. Remember, the physical, okay? The physical thing of both homozygotes and heterozygotes, okay? And for example, blood typing. AB blood means you have A blood and B blood, as in the proteins from them. So we have multiple alleles. Now, many genes have more than 2 alleles, meaning more than 2 phenotypes can be observed in the population. As in, look at the eye colors. You see multiple, like this is green and blue. You have a little brown. You have here. And it pretty much changes. My eye color is about like this, and sometimes it can, during the winter time, it looks more like this. But my dad's eyes are like this, and my brother's eyes is like this. So it's possible to have multiple alleles, like hair color. Is your hair just straight black? Is your hair just straight blonde? No. Some of us has different pieces in it, like different little, like natural highlights. That's what we call it. So this is a new allele that can be formed at any time when a mutation occurs. Because you can always, you ever seen people like just born with like black hair, and then when they got older, it start looking like they had like maybe little red streaks or something. This is because it was a mutation that occurred, and we already talked about it with a nitrogen base within a gene somewhere. So here's an example of like, here's some guinea pigs. Like obviously they most supposed, I guess they're supposed to have been black. But look at this guinea pig. If you saw this guinea pig and all of those, you'd be like, that ain't your mama and that ain't your sister and them. Yeah, you're gonna look at him like he's lost. And that is because it can be a mutation, but this also again comes into having codominance, as well as incomplete dominance. This is more your incomplete. It's just a mixture. We don't know. So here we go now with the codominance and everything for the humans. It's because of our blood type. Do you know what your blood type is? If you don't, you need to know. But there's 3 types of blood alleles that we have. We have A, we have B, and we have O. And a lot of times it's written as I, so it's not going to be, and it'll end up looking like a little line. But these right here are I's. Okay, so whenever you see this on the test, just know that that's an I for blood. They chose I for whatever reason. So human blood types are determined by the presence or absence of certain proteins on the red blood cells. So this is why we would have A blood, B blood, AB blood, or O blood. So the other ones are blood alleles, which either can be A, B, or O. But now these are actual your type of blood that you would have. So why is it important? I told you you need to know. It's necessary for you if you ever need a blood transfusion. Okay, because you see this would happen if you try to mix, if you have A blood and you try to mix it with B blood, your blood's going to clot immediately and you're going to die. We don't want that to happen. We want you to live. So red blood cells are incapable of blood, and it could clump together causing death. We need you to live. Also, it's helpful for cases like even when it comes down to parental disputes. We have to get blood as well because blood lets us know, hey, we got the same blood type, so it's a greater possibility that I'm your father or mother. And then we do the DNA swaps. So for instance, if you are a child with AB blood, whose mama has A blood, can't have a daddy who have O blood. That ain't your daddy. Like there's no possible way. So that's how they always do DNA tests. Do a little bit of blood, do a little bit of swabbing, and we can find it out. So how does this work? Well, each allele codes for a different protein. So that's your key thing again. Each allele codes for a different protein. And where is it found out? Remember, this is on the surface of the red blood cells. So A, remember A. Remember little I means it was O blood. So A is dominant. B is dominant. AB is dominant. And O is obviously, O blood is recessive. Okay, And in order for you to get O blood, your parents would have to carry it, or they would have to be O blood themselves. So the person who can give, like me, I have O positive, so I can give to anybody that I desire to give to. Okay, I can give to any one of you all. If something happened to you, I can donate to you. The only person who can receive anything is person who has AB blood. So if you have AB blood, you can get blood from anybody. So this is how it works. As you see, we have A. If you have A blood, you have an anti-B and you have an A antigen. B blood, you have anti-A, you have a B antigen. If you have AB blood, you don't have an antigen, so you're pretty much can accept anything. You don't have no antibodies do it, but you do have both antigens, so you can accept. And then O, we don't have any antigens, so it doesn't block me from giving it to anybody, but I definitely have a blocker that would not allow me to accept it from anybody. So possible genotypes. So remember, genotypes is stuff that you can't see, right? So in order for you to be A blood, you can be AA or AO. So I'm gonna write it right here, AA or AO. That means you are dominant A, or you have A blood. That's your phenotype is going to be here. Okay, these are your phenotypes. Remember, that's what we see or what we identify. These are your genotypes, all these letters. So if you're AA, you're A blood regardless. If you're AO, you're A blood regardless. But you carry an O gene, and that O gene will now show you how to, in order for you to have a kid with O blood. If you have B blood, you're either going to be BB or you're going to be BO. So every time you do a Punnett square to figure out what type of blood type it is, if this says, hey, a mother had A blood and the son had, the baby daddy had B blood, you need to do AA times AA and AO times AO and BB times BB. You have to do a lot of Punnett squares. It could be up to 4 or 5 Punnett squares per trying to figure out what the answer may be, because it has a dominant and a carrier. If you're AB, obviously you're just going to be AB blood. And if you are O blood, you're just going to be O. That means you got OO. Your base, that's what you are, OO. Best way to look at it is here is a square from here. If your mama is A blood, and your daddy's A blood, you will be A blood, AA. I mean, there's different ways, especially if they're carrying. But in order for you to be O blood, like me, you can either have the recessive gene from your mom and the recessive gene from your dad, or both of them are O blood. So I'm going to do an example for you because you have to understand how to do this. So a woman with A blood is claiming that a man with AB blood is the baby of her father, who is also AB. Could this man be the father? Well, a lot of us are like, yeah, because he AB, the kid. Not necessarily, so let's try it. So I'm going to do the woman. She's A, but I'm also going to do a Punnett square that says she's AO. Why did I do that? Because it didn't tell me. They just told me her phenotype. It never gave me the genotype. So because of that, I need to make sure I'm able to do both genotypes to figure out. AB won't never change. Okay, so let us write it together. So I'm just going to write his letters over here, and you can see. All right, so now I'm writing the mother. So A and A is AA. So this child, obviously, genotype is A, but the blood type is AA. AB, so that's an AB blood here. AB. AA. So this is A blood. AO, this is still A blood. That's why I'm writing those A's there. AB, this is AB blood. And then BO is B blood, regardless of if it has an O. So it's just asking you, could this man be the father? Yes, regardless, he's probably the father because he has it at least 3 times. He's on there multiple times. So we're not worried. If he wasn't on there at all, then no. So yes. So polygenic inheritance. This is when many human traits are determined by polygenic. So if you look here, this is like showing you like the phenotypes of skin. You can cross people and actually see what color the kids and stuff could possibly be, and what kind of genes. Any trait controlled by 2 or more genes are located on the same or different chromosomes. So as you see here, you see all those letters, all that makes up just your melanin. And it's just on, again, can be located on the same or different chromosomes. Example, eye color and skin color. A lot of times, the darker you are, the darker your eyes are. The lighter you are, the brighter your eyes are. It just kind of works together. So did you know, though, that internal and external factors can actually affect the inheritance and turn genes on and off? So, for instance, sea turtles, their sex and stuff can actually be determined during like the temperature of the ground. Siamese cats here, gene can be for dark fur and can be turned off by temperature and turns back. And that's why the ends of the tail, that's why they're darker here, because they can actually change little colors. So again, this is what I just stated earlier. So I just wanted to review. So this is just using the words. So this is hetero. So again, this is heterozygous. This is homozygous dominant. This is homozygous recessive. So which one is heterozygous? Again, this is hetero. So hetero always means different. This one's different. Which one is homo dominant? Dominant, yes. And which one is recessive? That one, because it's homo. All right, so determine the phenotype if blonde hair is recessive to brown. So if blonde hair is recessive, that means blonde hair is little b and brown is big B. So which one would this one be? That's going to be brown hair. Which one was this one? This is brown. Remember, dominant always wins. So what color hair is this? Blonde. So the cross of unknown genotypes with the individual of known genotypes is the test cross. And what type of blood is universal donor? Oh,